that we won't get very far into membrane functions if we do. When we get back next Wednesday, we'll definitely do membrane functions. We should start tissues as well. Next Friday, so next week, we'll have your next quiz, quiz number 3, and not sure the topic, but probably covering organelles and maybe membrane functions, something like that, and we'll continue tissues on that day. Your first exam is only 2 weeks from today, so it's 2 weeks. It's on a Wednesday, 2 weeks from today, so not too terribly far in the future. It's a little ways off. And that is the schedule. Do you guys have any questions for me about the schedule?
Today's date.
The 27th.
Or anything else about the course?
27.
Yes?
What's the on the first class? What will you go from
It'll start from the very beginning of what's on the syllabus, introductory material, all the way through biomolecules, cells, tissues. Anatomical terminology will not be on that. It will be on the second exam. So it's what the syllabus has, with the exception that anatomical terminology will be pushed off to the second exam. Otherwise, your syllabus should be accurate. Anything else? Okay, last time we were talking about organization of the cell, talking about cell membranes, organelles, nucleus as major regions. We were on the cell membrane. We had mentioned about different kinds of molecules that make up these components of the cell membrane. A lot of proteins, a lot of lipids, at least in the form of phospholipids, some cholesterol, carbohydrates as attached to various lipids or proteins. I did talk about carbohydrates, didn't I? Or is that on your list? Did I mention what carbohydrates do? And cell identifiers. Okay, good shape there on that one. Want to talk something, concept I want to call the, we're going to call the fluid mosaic model. And to understand that, we would have to, let's imagine just for a minute, we take a look at one phospholipid molecule. I've marked that one with a little green circle right here, the head or the polar end of one phospholipid molecule. If we looked at that in a microscope, presumably we could identify individual molecules in a really high quality microscope, and we can watch that in real time. We might mark that with our eye, look at that, turn our head away and look again and find out that same marked molecule is now over there. A second later, it might be over there. What this is saying is that these molecules are dynamic. They are not fixed in position. They are not in only one place. They actually move around. The proteins can move around. They may move from the inside to the outside, from the outside to the inside. This concept we call the fluid mosaic model. The fluid part of it indicates that the molecules can move. This is not a static membrane. It's not like a plastic Walmart or Kroger bag made up of all the same thing where the molecules are more or less in position. The molecules can move around. And in fact, the whole membrane has been likened to the consistency of olive oil, maybe, or something like that. But the proteins move around. The phospholipids move around. It's dynamic. That's the idea of fluid aspect of it. Not just that it's a liquid, but the molecules move around. Mosaic. What is a mosaic? Anybody take an art class? Got a pretty mosaic picture. And we see an entire picture, but it's composed of different elements that are strung together or pieced together to make a bigger picture. So we've got this nice picture, and I think it's made of ceramic pieces or maybe dyed glass pieces. We've all got all these different colors of glass pieces, but they're put together in a certain way as to make a scene. In our plasma membrane, it's not all one kind of molecule. It's a mixture of different kinds of molecules. It's a mixture of proteins and lipids. And these proteins and lipids move around, hence the idea of fluid mosaic. I guarantee I will ask you about fluid mosaic at some point on a quiz or test. I'm not sure if it's going to be a quiz, not sure if it's going to be a test, maybe it's going to be both, but I'll ask you about fluid mosaic at some point on a quiz or test, guaranteed. If you understand the concept of fluid mosaic, it's going to help you understand how a cell works, and especially how materials move across the cell membrane, plasma membrane. Quick overview of functions of proteins. I have hinted at these already, but maybe not clearly outlined them. Your textbook describes a lot of different things they do, and we're going to go into some of these a little bit more detail later. But as a quick overview, if we try to summarize a number of the different concepts portrayed in your textbook in this series of pictures, one of the things that we see proteins important in is transport across membranes. That is, they're very important in assisting molecules moving from the outside to the inside, from the inside to the outside. And that's what several of these groups of big yellow protein molecules do, or are doing on this series of pictures. Another thing they are heavily involved with is communication, especially if they've got carbohydrate tails on them. They're involved as receptors, detecting things going on around them, responding in a certain way, maybe giving a signal to the inside of the cell. They function along with the carbohydrates as cell identifiers. And the third thing I'm going to put here, just very simply, as structure. Some of these, and that's this very last next to last panel here on the right, some of these link toward various filaments and fibers and elements inside of the cell to give it shape and position. Some of them link cells together. They extend to the outside of the cell and bind cell to cell to cell to keep that cell in one particular place. So some of them do that as well. Oh, I've already got it written up on the screen. I didn't even realize I do that. Identity would be communication that's relevant to that. Let's start looking at organelles now next. We're going to go through a number of different organelles, not all of them that are in your textbook. So if I don't go through an organelle, if I don't cover it on the PowerPoint, that's going to be one that you're not going to be responsible for in a quiz or test. I'm going to pick what I think are the most critical ones, the ones that we're going to reference likely later on in the course. Some of the organelles include something called a cytoskeleton. Cytoskeleton are a group of proteinaceous strands, filaments, fiber-like structures. They extend in various areas or various directions of the cell. Some of them are very important in structure, providing a framework. Maybe you've seen a large building being built and you see the steel beams and girders and cross pieces that form the framework that then floors and walls and ceilings are attached to. These filaments, these various kinds of cytoskeletal elements can be involved with that. Others are very important in movement, moving things around in the cell. They may act kind of like a rope. It's a big long filament, something gets attached to it, the rope gets pulled, the filament gets pulled, and it moves it from one end of the cell to the other. Maybe this might be the way chromosomes get moved around during cell division into daughter cells by various kinds of these cytoskeletal elements. They're often divided in some different sizes. We've got the smallest ones called microfilaments. An example of this one that we'll see later, so this is not the only kind of microfilament, this is just one example of that. But an example of one that we'll see later when we talk about muscle contraction is actin. Actin's involved with muscle contraction. An example of an intermediate filament is a structural one, keratin. Keratin provides structural shape in our epidermis of our skin, and also things that develop from the epidermis of our skin, hair and nails, for example. We'll talk more about that when we get to the integumentary system, so we'll see this one later on. The thickest ones are microtubules. They're composed of a protein called tubulin. These are relatively thick ones. They may be involved in either structure or movement, and they're components of other things that we're going to see later as well. Microtubules are one of the structures that helps to move chromosomes from during cell division from a middle point to be pulled apart into the separate daughter cells. Structurally kind of related to cytoskeleton, particularly those microtubules, are some structures called cilia and flagella. What is common about these? These are projections to the outside of the cell. They both project to the outside of the cell, and they're both composed of microtubules. There's a certain pattern in which they're arranged that's common to both. So in many ways, they're very similar. They are composed of microtubules. They project the outside of the cell. They can move. They wave around. So what's different between the two? See if you guys have been keeping up with the reading, or might just happen to know this one off the top of your head. It's our first clicker question of the day. Cilia and flagella. What's similar? Composed of microtubules. They move. They project the outside of the cell, but there are some differences between the two. One of these is going to be typically in a situation where it's relatively long, but there's just one present. Other situations, one of these, either cilia or flagella, will be in a situation where there's typically a lot of them on a cell, and they tend to be relatively short, comparatively short. Okay, let's see what you guys picked on this one. How many might be able to figure this out on your own? Most people picked B. B is short and many, and that's correct. So cilia are usually present as shorter structures, and there's usually a lot of them. We might have them perhaps in our respiratory tract, tracheum. We'll learn why they might be there. Well, briefly, they're going to be sweeping some dirt and debris out of our respiratory system. They can move materials over their surface. Flagellum. Flagellum is pretty rare. We only see that in one human cell, and that human cell is sperm cell. They have a little bit different mode of movement. Flagella here at the bottom kind of move in an undulatory fashion, and that is they propel the entire cell. They allow the sperm cell to swim. Cilia don't cause their own cells to move. The cells are usually fixed in position, and so they sweep material over the surface unidirectionally. In our respiratory system, they sweep up mucus and dirt and debris up and out of the respiratory system. We'll tell that story when we get into tissues a little bit later. Any questions about cilia or flagella? Also kind of, oh, I forgot about this one. Some cilia do not have a purpose that is locomotion based. Instead, they are sensory. And we won't talk a lot about this this semester because this is more with sensory organ systems in the second semester. But some of them, when bent from side to side, send a nervous system signal. They basically detect movement. And this is, we use these mechanoreceptors in our inner ear region for detecting sounds or balance or equilibrium, and actually in a few other places in the body as well. And we'll talk about that a little bit later. But realize that some cilia can be sensory as well. Basically, when you deflect them, it sends a particular kind of signal. Next kind of organelle is also structurally related to the first two, and so these are called, this particular organelle is called a centrosome, which has a couple of short cylinders associated with it called centrioles. Maybe you've heard of centriole instead of centrosome. So they are two little short cylinders arranged at right angles to one another. They are composed of microtubules as well. So maybe they look just like what you saw with the cilia and flagella. They look like little short segments of cilia and flagella. Function, microtubule assembly. So again, they're linked structurally, functionally with some of the last two things, particularly with microtubules. These are made of microtubules. This pericentriolar, this darkly staining area around it, has lots of little kind of formed elements of joined microtubule or microtubulin molecules. And apparently this is where microtubules get assembled. These appear to be relatively important during cell division. When we have that spindle apparatus forms that pulls the chromosomes apart to respective cells. this tends to be pretty active in organizing that. And it has to get replicated for cell division to occur. Okay, quick bit there on centrosomes. Don't have a lot to say on that one. Ribosomes are next. First of all, what do they look like? Ribosomes look like little tiny spheres, little tiny dots. Maybe you can just barely make out where you're at. These little tiny, looks like they're bluish dots all over the cell. They're all over the inside of the cell. Some of them are free in the cytosol. Some of them are stuck to other organelles. Ribosomes. If we look at them, they're made up of both RNA and proteins. There's some subunits that are kind of stuck together there. But they look like little dark spheres, little dark dots or so. Function of ribosomes. And that's going to be our next clicker question. Okay, I have to tell you something first of all. Ribosomes, what they are, their function, they're places where proteins are assembled. So protein assembly, protein production occurs at ribosomes. If a cell has a lot of ribosomes, it's producing a lot of proteins. Now I've given you enough information to actually answer this question. You have to go back to that central dogma idea and see, what do you think they're involved with? Is it replication, transcription, or translation? And again, I've given you a tip here about they're involved with protein production. Ribosomes are a place where proteins are produced or assembled or made. See if you remember that from your previous notes from last time. Trying to reinforce what we learned earlier. And you could just turn your page back on your notes. You should have your notes from last time and see where proteins are assembled. Remember the DNA to RNA to proteins? Just take a look back there. C is the correct answer, but you should have had a little higher percent going on there. Remember, translation is a process by which information from RNA is used to make proteins. So in this case, translation is what occurs at ribosomes. Endoplasmic reticulum is our next organelle, sometimes abbreviated ER. Endoplasmic reticulum looks like a series of membranes and channels and folds and pockets. It's a highly folded membrane forming canals and passageways and cavities and chambers. We use the word canal here, and so think about maybe a function of canals like a waterway canal, like maybe the famous Suez Canal in the Middle East. Functions of that would be, why do we have canals? Panama Canal, Suez Canal? Transport, yeah, that allows us to move ships from one place to the other, allows for transport. And that is kind of what's going on here. There's some production of some biomolecules that occur in these various places, and also they are transported through these canals into various areas. Now, what is being produced varies on the kind of the ER. One kind of ER is rough ER. I haven't got the clicker going yet. One kind is rough endoplasmic reticulum, and one kind is smooth endoplasmic reticulum. Rough ER has ribosomes attached to that. Smooth ER has no ribosomes attached to it. So if we look over here, we may be able to see some rough ER. If you can maybe make out the blue dots on this greenish part of the membrane, that's the rough ER. And here, the smooth ER has none of those blue dots. There's no ribosomes. So we distinguish between rough and smooth ER based on their structure, their anatomy. They have a little bit different functions as well as a result. And I bet you can probably figure that out now with something that we've already said. So now we'll start our iClicker answer possibilities. Maybe you've already thought about this now and you can answer this one pretty quickly. You might be thinking about functions. Now we said an ER in general has to do with production and transport, but of what? and the what varies a little bit in smooth and rough ER. If you looked at a cell in a microscope and it had a lot of rough ER, you might conclude that it produced lots of, what might be a best choice here? Okay, you've had enough time to try this one, and your choices are, oh, very good, yes, you remembered that ribosomes produce proteins, that's a site of protein production, and now, so with rough ER areas, you tend to have a lot of protein assembly production, delivery, that happens in rough ER. Well, what happens in smooth ER? Smooth ER, one of the things that can happen here is a little bit of lipid production. Lipid production often occurs more in smooth ER to a certain degree.
Does it make calcium too?
Doesn't make calcium, but you're probably thinking there's a form of smooth ER called the sarcoplasmic reticulum that's important in muscle cells, and it stores calcium associated with muscle contraction. I bet that's what you're thinking of. Does that sound familiar, sarcoplasmic reticulum or something? We'll get to that later on, so don't worry, you don't have to know sarcoplasmic reticulum right now. But we will get to that a couple of units down the road. It's a form of smooth ER. It doesn't make it, but I guess it stores it or regulates flow in and out of it pretty heavily, yeah. Lipid production, and my next thing up here, oh, why did I put a bourbon bottle up there? It turns out that smooth ER also is important in detoxification. That's to remind me I'm supposed to talk about this, and about the avenue of drug or alcohol tolerance. Okay, I know no one here has drank any alcohol yet, but if you did, you might be it's kind of a joke there. You might be aware that there's a phenomenon. If someone has been using a particular drug or alcohol, or maybe consume a lot of alcohol for a while, one might develop a little bit of a tolerance to that. What's going on there? Why does that happen? It takes more of the alcohol to produce the same desired effects. Well, detoxification does occur in smooth ER, and your cells don't always have a fixed amount of any of these organelles. They can respond to environmental challenges and change. There's a lot of smooth ER in your liver cells, and maybe you know the liver is a place where alcohol is detoxified. It is a toxic material of the body. It has to be gotten rid of. It's detoxified in the liver, and it does so in the smooth ER. Well, when faced by a lot of this toxic materials, your body responds by making more smooth ER. So those individuals who are relatively resistant to a particular drug, toxin, or maybe alcohol, their body's adapted by growing cells or getting more smooth ER that has more smooth ER in their cells, tend to be a little bit more resistant to it. Now, I'm not telling you to go out and drink a lot of alcohol so you get a lot of smooth ER so you're protected from it. If you do that, there's a lot of other toxic effects that'll kill you or hurt you long before it'll overweigh the advantages of having that. But anyway, that's the way that drug tolerance or alcohol tolerance works. Emphasizing the fact or the idea that we see this idealized cell, but none of the cells in your body look exactly like this. They have varying amounts of these different kinds of organelles. And they can even adjust that to meet different needs. And this functional anatomy is strongly correlated to structure, or the structural is strongly correlated to function. Questions so far? Next organelle is something called the Golgi. It may have an appending word, Golgi apparatus, Golgi body, Golgi complex. We'll just call it Golgi here because that end word can change quite a bit. But Golgi, what it looks like is a series of flattened membranes or flattened sacs. People have described it as looking kind of like a set of stacked pita bread, perhaps, where we've got these little flattened cavities or flattened chambers that are all stacked up one on top of the other. And has a very distinctive kind of arrangement associated with it. Golgi can act a lot like a post office or a FedEx office. Oops, we'll tell you that story in a minute. Post office or FedEx office. It receives proteins that have been made from ribosomes, rough ER, and it tends to modify them, maybe package them, and then label them to be delivered to a certain destination. Maybe to stay in the cell, to be incorporated in the cell membrane, maybe to be exported out of the cell. It modifies the proteins in a certain way, maybe makes them functional. This might like be the post office. You take a package over the post office or FedEx office, and maybe it's not quite ready to go. You still got to put it in a box somewhere. Maybe you have to purchase a little box to put it in, or the right kind of mailing envelope or package. And then it has to be from there, it's going to be stamped or modified as far as delivery, where it has to go. And it's sorted in some way. Some of them, maybe it's mail that goes out of town versus in town, or wherever, or maybe it's air freight versus ground freight. It's got a various kind of things that might happen in a post office. And kind of the same way with the Golgi, an area where proteins are modified, sorted, packaged, labeled, and maybe even labeled for specific delivery. And a little bit more about that a little bit later. We'll get into that just a bit more. But I do have another picture up here, and this is a picture of a baby with a disease called cystic fibrosis. This is a genetic disorder that is characterized by individuals producing too much mucus in various areas of their body. The mucus tends to build up in some ducts. It can cause kidney problems, particularly respiratory problems, and that's what this person is doing here, or at least in the cartoon version. They're kind of tapping on the baby's back to loosen up some of the mucus that's down the lungs and the respiratory system. This is there is no cure for this particular disorder, although we've got better treatment. It used to be very few individuals lived past about 15 or so, and now most individuals live maybe into at least in their 20s or 30s, but still a difficult disease to treat, and certainly there's no cure for it. The reason for having this disease, or its problem, is actually traced with one faulty step in the Golgi complex. There is a protein that needs to go to basically to the cell membranes, and it doesn't get basically modified in the right way in the Golgi complex, and so it's a non-functional protein, and you end up getting mucus buildup on certain areas of the body. I might have another picture of that. Nope. I think I have a picture on maybe further down the PowerPoint. Maybe that's what I'm thinking of. I'll show you a picture of that protein and the dysfunctional aspect of it later. But again, that illustrates that very severe illness that is fatal eventually from one step in a Golgi complex. It's not even the Golgi's missing, it's just one faulty step in the Golgi complex. It's genetic. You get that one inherited. Likewise, this next disease, which is more common in individuals of Jewish descent, Tay-Sachs disease, it's another inherited disorder. Individuals get neurological symptoms very early, typically blindness, inability to move, motor movements. Babies generally never learn to walk associated with this. And death usually results age 1 or 2 or 3, certainly in the first couple of years of life. Neurological disorders happen very quickly. Another inherited disorder, another one with no cure or no effective treatment. And this is due to a faulty aspect of another organelle called lysosomes. Again, trying to illustrate that one little minor problem in some of these organelles can have far-reaching effects on the health of individuals. Well, what are lysosomes? First of all, before I explain what happened here on this particular disease, lysosomes are bags, they are spheres, they are containers containing strongly digestive enzymes. Now, what I mean by digestive is not food digestive. I mean enzymes that break things down. Strong, maybe corrosive might be the word. These contain enzymes that are very strong. They break things down. Why would you want to have such a dangerous brew of materials inside one's own cell? Well, they're used to recycle extra molecules that build up too much. They need to be broken down. They can be recycled with component parts used to build up something new. Old, worn-out organelles can be broken apart this way. They may contain things that would, say, break down an invading pathogen like a bacteria. White blood cells can use these to break down bacteria, for example. So there's a number of different uses, and we normally have to have them to keep bad things from building up. And that's what happens in Tay-Sachs disease. There's one faulty enzyme associated with a lysosome. and that enzyme doesn't break down a glycolipid. And so there's a glycolipid in the cell membrane that builds up in these neuron cells. These are neuron cells. This glycolipid builds up, and that eventually causes the neurons to dysfunction, and thus you get all kinds of nervous systems, kinds of pathological symptoms. You get mental retardation, you get lack of movement, cognitive abilities, blindness, and so on, that result of that disease. Questions about lysosomes? Next kind of organelle is mitochondria. Mitochondria are relatively large organelles that are somewhat elongate, often been described as sausage-shaped. They have a unique double membrane associated with them, probably an endosymbiotic origin. Probably they're captured bacteria or other prokaryotes is the origin of these particular things. Everybody remembers midi-chlorians from Star Wars. Yeah, remember when they're taking midi-chlorian counts back maybe episode 4 or so, that living life force, endosymbiotic living life force in all of them. That was George Lucas's idea of mixing mitochondria and chloroplasts, which are endosymbiotic. Anyway, moving on from that. Mitochondria, double membrane, they have their own DNA, again, probably from that endosymbiotic origin. They can self-replicate. Major function of mitochondria is?
ATP.
Yes, this is a place where ATP is produced. Not the sole place where ATP is produced, but most of the ATP production occurs in mitochondria. Talk more about these when we get to muscle tissues and energetics and exercise and fueling muscles for energy. They come into particular play. There's some interesting things there. We'll talk more about them. But if you know for now, again, they have this double membrane. These are sites of ATP production, sites of energy production. That's pretty good for now. You need that ATP to fuel all these kinds of chemical reactions that we've talked about. A lot of the energy produced comes by moving various molecules, mainly hydrogen ions, across this inner membrane. So I've got a question here. This is kind of a thinking question just for a minute, but it gets at a concept that it will permeate the whole semester and will appear on your final exam as a comprehensive question. But if we look at a mitochondria, it has an outer membrane, and then it has a looping inner membrane. The inner membrane is highly folded, and this is where most of the energy is produced by shuttling molecules across this inner membrane. That's how most of the energy is produced. It's an anatomical configuration that is better than this way of producing inner membrane. So here's an alternate scenario, which we don't have for mitochondria. It has an outer and inner membrane, but the inner membrane is not folded. The top one, the inner membrane, is folded. Why is this a better anatomical configuration for producing energy than this one? Any idea?
Produces more friction between the molecules.
It's a good idea. Not quite right here in this one. Looking for a different answer than that. You're getting closer.
It's to get the ions to go back. Oh, gosh.
I heard something maybe in the back, maybe?
More space.
More space. We might think about which of these inner membranes is bigger, this one or this? More surface area.
Okay.
This is a bigger membrane. You could pack more membrane in by highly folding it and looping it compared to this way. By increasing the surface area, we can move more ions across it and generate more energy. Surface area is going to be an important theme throughout the course. We're going to see surface area important for absorption of nutrients or movement of gases from oxygen from one place to the other, maybe lungs to the bloodstream or bloodstream to tissues. We're going to see surface area as a relatively important theme. And don't be surprised if you see a surface area question on the comprehensive portion of the final exam. Again, we're trying to link structure and function here. Think about these as related kind of topics. If you're just trying to memorize a lot of things in the course, it's a very difficult way of doing it. But if you're learning why things are the way they are, it makes it much easier to learn. All right, so where we've been so far, we had this list earlier. We've been through plasma membrane. We've been through the cytoplasm with all the organelles. And now we're on nucleus, our last organizational region of a cell. Nucleus, it's that large big sphere in the middle of the cell. Usually there's one, but some cells don't have a nucleus. Red blood cells, at least the mature form, don't. Some of them may have multiple nuclei, skeletal muscle cells. There's one kind of bone cell that's got a bunch as well. So it's not always just one, but for many cells there's just one nucleus. What's important about the nucleus? Nucleus is where DNA is located, except during cell division. DNA is contained in that double membrane that forms the cell, that forms around the nucleus. Nuclear envelope is that membrane that encloses the nucleus. It's kind of an unusual arrangement again, which has some interesting ideas about evolutionary origin, but really beyond the scope of this class. Nuclear envelope is the nuclear membrane. That's the membrane about the nucleus. One way that it's constructed differently as compared to a plasma membrane is that there are large holes in it. And we call those holes nuclear pores. Nuclear pores are the large holes, large openings that are inside, that are in that in that nuclear envelope membrane. What do you think those openings are going to be used for?
To get the RNA out into the cell to the ribosomes.
Just like you've got a door in this room, allows things to go out or allows things to go in. And it's a larger opening, so it allows particularly large things to go in and out. As mentioned before, what may go through there are large proteins or RNA that's being made. RNA is made in the nucleus from the DNA. Ribosomes are structures that are made inside the nucleus as well. So there's some relatively big things, I'm using kind of a broad term here, molecule structures that need to move through that membrane, and those openings allow that to happen. Somebody mentioned RNA there, and one of the places where RNA is made, at least in the form of ribosomes, is something called a nucleolus. Nucleolus, tomato, tomato. At any rate, this is a dark area that is in the nucleus. It looks like a dark area in the nucleus. Its function is ribosome assembly. Cells which have a dark, distinct nucleolus are often involved with a lot of protein production. Because to make a lot of proteins, you need a lot of ribosomes. To make a lot of ribosomes, you're going to have a large, distinctive nucleolus. Again, function is complementing structure. All right, so what we've got a nucleus. You should have something about site where DNA is at. Nucleolus, site of ribosome assembly. The envelope is the membrane, and the pores allow for large materials to go in and out, mostly to go out. Ribosomes are generally going out. Messenger RNA is going out. There can be some proteins and some other things that go in as well, but mostly for things to go out. Questions at this point? We're going to start putting all this together now, some of the different things that we've learned. So we're going to draw a picture. This is a cell, and this is the nucleus. And this is our central dogma. Where would these events happen? Where would DNA be made into RNA, and where would RNA be made into proteins? Where do you think we should, if we tried to put, say we wanted to write this up here in our new picture, DNA being made into RNA. Where do you think we should put this now in our cell picture?
Nucleus.
In the nucleus. Very good. So here's the nucleus. Here's that one step. We've got DNA being made into RNA. Depends on the kind of RNA. It could be ribosomal RNA, or it could be messenger RNA. I know those terms may not be familiar to you. But regardless, it happens there because the DNA stays in the nucleus. And that RNA now has to move out, so we're going to make a little opening here. It's the RNA is, here's our RNA, it's moving, you can't read that. RNA is now moving out, that's my little dotted line. It has to move out through those nuclear pores. And now it might go somewhere. Where do you think it might be going? Maybe we need to write another structure here in our cytoplasm.
Endoplasmic reticulum.
A particular kind of endoplasmic reticulum.
Rough.
Rough ER, where there's ribosomes. So it's got to go somewhere where there's a ribosome. So now we're going to draw a ribosome in here. And right here we've got RNA is used to make proteins. This is our site of protein assembly. Maybe we get a protein that's being made. Maybe it needs to be modified, sorted, packaged in some way. Let's add another structure where that might happen. What do you think that might be?
Golgi apparatus.
The Golgi, yes. So maybe we can draw a little Golgi apparatus, a couple of series of flattened things here. And from there, a modified protein can go a lot of different places. Notice we're kind of putting together all these different organelles. They don't exist in complete separation function. And here's, well, this is a picture that's kind of showing some of this. Here's our DNA being made, or being used to make RNA. RNA leaving out the nuclear pores. It's going to a ribosome where translation, that is protein production, is going to be occurring. Let's see here. Transcription, just to review, on a clicker question. Information from DNA is used to make a strand of, right on the board here. RNA, yes. So transcription, remember, is what I've just drawn here. This arrow, that was transcription. Information from DNA used to make a strand of RNA. And I don't expect you to know the details of how this is made. If you know this occurs in the nucleus, if you know that transcription is a process by which RNA is made by or made from information in the DNA, that's good enough for this course. We won't get into the details of how that's made. Oh, sometimes I use this word, messenger RNA, mRNA. This is how messenger RNA is used. Translation, remember, is a process by which messenger RNA is used to make proteins. There's this little section, this little string of messenger RNA, and it basically gives directions on which amino acids need to get strung together and which order they do. And so basically what happens, we've got ribosomes, and there are some other things that come into play, other kinds of RNA to come into play. And what this is showing in the big picture is, this is our messenger RNA. This is the instruction booklet here. This is the instructions. And it's saying what amino acids need to be strung together. Maybe we're going to string this one, whatever this green one is. And it's strung together. Then we're going to add this red one. And then we're going to add a whole bunch more until we get our entire big protein string of amino acids. So we have this messenger RNA, these instructions, and it is used along with some other forms of RNA to assemble these amino acids together and make a viable protein. What's my next thing here? Oh, okay, here. Let's see if you can do that now. Can you make up your own sentence right now using those 4 words to explain what happens in translation? See if you can do that on your own paper. If you can, you probably understand it. Note there's not necessarily one right way of doing it, but can you use those 4 words to make a sentence to explain how translation works? Quick overview. And I know I went through this quickly, and that's why I'm asking you to see if you can try to do this. When you're studying, do these kinds of things. Use writing as part of your study. Try to write out an explanation, a shortened explanation. Try to restate things in different words, not just use the exact words that your textbook uses or that I used in class. See if you can write a sentence. If you come up with a sentence and want to share, anybody have a sentence they'd like to share? An idea? I bet you came up with something. Or maybe you could look off your neighbor's paper and read what they wrote down on their notebook. Would that be okay? This is a learning process. I don't expect you to get it just right. maybe a lot of people will get it okay. You got an idea? Okay.
The mRNA goes through the ribosomes, matching up amino acids to make proteins.
That sounds pretty good. That sounds like a pretty good description right there. That's not the only kind of description that you could use. I got up here 4 different ways you could describe that. There are more different ways. And you can get this from the PowerPoint later on Blackboard, so there's no reason to try to copy all 4 of these down. The point of this is the idea of understanding in the course. Don't just memorize the definition. Here's 4 different ways you could describe how translation works using those 4 words. None of those are more right or less correct or more wrong than the others. Just different ways of doing it, different ways of explaining that same concept. And this is what I want to encourage you to do, to make sure you write, to try to understand, to restate things rather than just rote memorization. I don't think I ever asked you just for rote memorization of a definition. Continuing with this idea of it's all connected, we've got a few minutes left. Oh good, cover a few more slides. This is the last stage of what we just saw. So you guys mentioned, somebody mentioned the rough ER. Here's the rough ER, protein bound in a vesicle going into the Golgi complex right here. It's being modified, it's being sorted, labeled, and it may be sent to stay inside the cell. It may be linked up to be incorporated in the cell membrane, plasma membrane. It may be destined to go outside of the cell completely. All connected. We've seen this before, and we remember the idea that some cells undergo mitosis regularly, some don't. Sometimes it is important not to reproduce. Sometimes it's important to reproduce at a particular rate. There are certain kinds of cells in the body that do regularly reproduce, and we call these stem cells. Stem cells, you're familiar with this or heard of this in the news maybe about stem cell. Stem cell controversy, should we be using stem cells? What are some characteristics of stem cells? Stem cells have great mitotic abilities. They tend to be able to reproduce, to replicate themselves, to divide very readily. So they can replicate, they can make more copies of themselves. They also tend to be undifferentiated, immature. What do I mean by that? Here's something you're going to see later on which describes how blood cells are produced. Now at the very bottom we've got our final cells or mature cells. Some of these are red blood cells carrying oxygen. Don't write this down. Some of these are platelets involved in clotting. The rest of these are various kinds of white blood cells fighting diseases. These cannot reproduce. These have very limited or no capacities for reproduction, for division, for replication. There are these stem cells that reside in various areas of your body, like bone marrow or thymus glands. This says pluripotent stem cell. It can ultimately form a red blood cell or a white blood cell or a platelet. But it is immature. It cannot transport oxygen. It cannot fight diseases. It cannot clot blood. It can do none of those things. Looks like we're out of time, and we're going to see how the concept of stem cells is linked to cancer, and we'll look at some disturbing maps about Kentucky on that day as well. So can't wait to do that day, because there's some fun things to talk about there.